Hi, my name is Evan Eichler. I'm a professor in genome sciences at the University of Washington. I'm also a Howard Hughes investigator here in Seattle. My lab focuses on dynamic regions of duplications and their role in both disease and evolution, and how they contribute to copy number variation. Where was I when the Berlin Wall fell? Well, I was a third-year undergraduate student studying biology in the University of Saskatchewan in Saskatoon. I celebrated the day with a glass of champagne. And also there was great hope, I think, for our generation that this was the beginning of a new era of really a lack of repression and suppression of people. As a Canadian of German descent, it also meant more to me. This is an opportunity for members of my family, my grandfather's generation, to reunite from the East and the West, in some cases for the first time. So what was our breakthrough? Well, our breakthrough was the complete sequence of the human genome, really from end to end, or what we call telomere to telomere. This was for me a fulfillment of a task that I started back as part of the original Human Genome Project back in 1997. At that time, by 2001, most of the human genome had been sequenced, but about 10% remained unfinished. So over the last 2 or 3 years, a group of us called the Telomere to Telomere Consortium, led by myself, Adam Philippi, and Karen Miga, took the task to actually finishing these difficult regions of the genome for the first time. Advances in long-read sequencing made this possible, really to access these regions and traverse them. So it was driven essentially by this new technologies. The essential new findings from this work was our first complete sequence, where we had all representation of every base pair, at least in one individual, present for the first time. This provided fundamental new insights in terms of how genomes change and how they evolve. It included regions of the genome that are important for segregation, called centromeres. So the transmission or segregation of chromosomes faithfully during meiosis and mitosis, the sequence underlying those signals were identified. It provided insight into the organization of genes called the rDNA genes that are important for actually building the ribosomes within every living cell that are critical for protein synthesis. And perhaps most interestingly, it identified regions that contain genes that are unique to humans, that are not found in other apes, such as the chimpanzee and gorilla. These contain genes that are important for building a bigger brain, increasing the number of neurons, and increasing the number of synaptic connections. The benefits to the world are that the human genome is now complete, at least for one human. We have essentially a book of instructions. We're still learning how to read this properly and how to write this code, but it serves as a new reference for actually improving the discovery of genetic variation for many other human diseases and human traits. It also provides a better template for designing probes to actually treat essentially genetic diseases. And it really, I think, essentially reveals entirely new forms of human genetic variation for the first time, and regions that change much more rapidly than other regions of the genome. So what remains to be done? Well, now that we have determined how to actually completely sequence one human genome, the goal is to do this for many, many more genomes in the future, in an effort to really develop a pan-representation of how all human genomes are related to one another. What keeps me awake at night? Well, really, it's this duality of our genome, that the rapidly evolving regions that make us uniquely human and give us unique traits also predisposes to disease. And the fact that these regions are so non-randomly organized still puzzles me. And what's my hope? My hope is that the methods that we've developed will be applied to really characterization of all human genomes in the future, to really understand the genetic basis of disease, especially for diseases such as autism and developmental delay, where many families still do not have a genetic cause, even though we believe one underlies it.